What about long-term memory? When we give repeated tail shock, we activate the serotonergic cells more. They release more serotonin, increases the level of cyclic AMP more, activates the cyclic AMP-dependent protein kinase more persistently. So now the cyclic AMP-dependent protein kinase can do two jobs, not just one. Number one, it goes to the presynaptic terminal where it enhances transmitter release, as it did in the short term. But now it does something very interestingly. The catalytic subunit moves into the nucleus. So one of the reasons you need repeated training is to allow second messenger kinases to move into the nucleus. And there they activate transcription. They act on a transcription factor, and Tom told you what transcription factors are. They're proteins that bind to specific sequences in DNA. This transcription factor is called CREB, cyclic AMP response element binding protein. And it binds to a sequence in DNA called the CRE, cyclic AMP response element. When it becomes phosphorylated by pKa, it can bind to that sequence, activate genes, and these give rise to the growth of new synaptic connections. And it's the growth of new synaptic connections that is the maintained form of memory storage. This growth is what is responsible for the maintained strength of synaptic connections. And if you leave this room remembering Tom's lecture, you will actually have an anatomical change as a result of this.